quantity is one thing. You put enough blocks together, you can grow a massive amount of cells. But I think what's more important to us is the difference in quality we're seeing of the cells that are growing in our blocks and making these microtissues and these environments to maintain themselves, excuse me, versus growing them in a petri dish or another 2D format, is really giving us some novel insights into what is happening with how cells interact and behave biologically. But another thing that we're starting to dive into, and we have some papers that are going to be coming out later this year, but the preliminary results have already been really exciting, has to do with how cells communicate with each other and what's known as paracrine signaling. So cells are constantly talking to each other by sending these little biological packets known as exosomes and extracellular vesicles that contain proteins, growth factors, and a whole number of wonderful things between each other, and kind of telling them, hey, how to behave, or I'm over here, so you go check out the place over there, all these different things, and or how to repair a tissue. And I mentioned that the cells tend to be metabolically more active when they grow in our block because they're making their own environment, but they're also communicating more. And we've actively seen this, that compared to 2D, when they're in this sort of 3D tissue mimetic environment, they communicate a lot, they're healthier, but we're also able to collect and intercept some of those communication packets, those exosomes and extracellular vesicles, because of the nature of our microchannels. We can collect the media that they're in, and then we can characterize those biologics and see what's in them and what's going on,